Chapter 11, Classifying and Characterizing Prokaryotes. In this chapter we're going to cover general characteristics of prokaryotic organisms, modern prokaryotic classification, survey of archaea, and survey of bacteria. Now, because these first two are quite short, I'm going to put them together and put them with this overview. So, on to general characteristics of prokaryotic organisms and modern prokaryotic classification. The learning objectives for general characteristics of prokaryotic organisms are: identify 6 basic shapes of prokaryotic cells, list 3 common types of reproduction in prokaryotes, describe snapping division as a type of binary fission, draw and label 5 arrangements of prokaryotes, and describe the formation and function of bacterial endospores. For modern prokaryotic classification, the learning objectives are: explain the general purpose of Bergy's Manual of Systematic Bacteriology, and discuss the veracity and limitations of any taxonomic scheme. In this recorded lecture, we're going to cover the morphology of prokaryotic cells, reproduction of prokaryotic cells, arrangements of prokaryotic cells, endospores, and modern prokaryotic classification. Prokaryotic cells, most commonly, come in the following shapes. If they're round or spherical, we call them coccus. The plural is cocci. And then I'm going to skip down here to rod-shaped, which is bacillus. Now, short, chunky little rods, which can appear as if they're coccus when they first divide, or bacillus when they're getting ready to divide, are known as coccobacilli. E. coli is an example of a coccobacilli. Sometimes the rod gets a little bent. Sometimes it gets a whole lot bent. We call these vibrio, or curved cells. If the bacillus gets really long and starts to form a corkscrew shape, it can be either a spirillum or a spirochete. Spirilia are rigid, and oftentimes they have flagella coming out of either end. Spirochetes, on the other hand, are flexible, and they have endoflagella, which you may remember from previous chapters, are like flagella that have been ingrown. They're underneath a sheath, and they help the cell move by spiraling. There are other cells that are pleliomorphic. They don't have a cell wall, and so they can form a number and variety of shapes within the species. So we call this pleliomorphic. Archaea come in a wide variety of shapes beyond this, but they're rare. These are the most common types of shapes or morphology of prokaryotic cells. On to reproduction of prokaryotic cells. Prokaryotic cells, generally speaking, reproduce by binary fission. They duplicate their chromosomes. The chromosomes are separated to one side or the other of the cell, and then the cell wall pinches in until it separates, and you have 2 different daughter cells. Now, sometimes this pinching off is somewhat incomplete. When we get into arrangements, we'll talk more about that. But at this point, let's talk about snapping division. Some bacteria, once this cell wall has formed to separate the 2 cells, will rupture almost to the end, and you'll have what's kind of like a hinge. So one will flop to one side, one will flop to the other side, and they'll lie parallel to each other, even though they divided through the short part of the bacilli. We'll talk more about that in the next slide also. And the third type of reproduction within prokaryotic cells, which is unusual, is budding. That's where you get the smaller cell coming off of the larger cell so that you can tell which one's the baby. This is more commonly seen in yeast, but some bacteria do undergo budding. On to arrangements of prokaryotic cells. First, we're going to talk about the cocci. If you get a circle and you divide it in one plane, and this division of the cell wall, they have a tendency to stay together and form 2, we call diplococci. If they, once they divide, they're on their own, they're separated, we call those single cocci. There's a single cocci. But you can see that this organism has a tendency to form diplococci. Now, if you divide in two planes, first this one, then this one, then this one, then this one, and it has a tendency to hang together so that you form these two-dimensional squares or tetrads, that is also an arrangement. Now, if you divide in three planes and tend to stay together so that you form these cubes, we call these sarcina. Now, the last one for cocci would be staphylococci. That's where you're dividing in these multiple planes hanging together so that you form these large grape-like clusters. Staphylo is Greek for grape-like, so we get grape-like clusters. On to the bacilli. The bacilli have fewer arrangements because, generally speaking, we only divide through the short end. Now, if they divide and they remain separate, then we call them single bacilli. But if they have a tendency to hang together in these chains, we go with streptobacilli. Now, cocci can also form strepto or chains. That would be if they only divided in one plane and they hung together. So, strep, of strep throat fame, is a streptococci, but streptobacilli are fairly common. Now, here's where we get snapping division. If you have a bacillus that divides through the short end of the rod, but you have a hinge there after the separation of the cell, we either get these V-shapes or what are called palisades, where they're lined up long ways. Now, why am I making you learn all of this? Because this is a way to identify organisms. Certain genus and species of bacteria have typical shapes and arrangements. So, for example, diplococci. Neisseria has a tendency to form diplococci. It's one of the ways that we can identify it. Staphylococci of Staphylococcus epidermidis or Staphylococcus aureus tend to form these grape-like clusters, so we can use this to identify them. Micrococcus has a tendency to form tetrads. Corynebacterium diphtheriae has a tendency to form these V-shapes and palisades, and we can look within a stain and look at it, and that gives us a leg up on identification. Another structure or morphological characteristic that helps us identify bacteria are endospores. Also, from previous chapters, you know that it's very important to know whether an organism produces endospores so that you know better how to control them. So we take a look at endospores, we see whether they're completely round, they're more ovoid shaped, whether they have a tendency to form at the end of a cell, in the center of a cell. All of these are used in identification. Speaking of identification, now's as good a time as any to introduce you to Bergy's Manual of Systematic Bacteriology. This is the go-to publication for identifying bacteria, also for finding out what their nutritional requirements are if you're trying to grow them in the lab. No microbiology lab should be without a Bergy's Manual. Having said that, it's not the complete authority on identification and classification of microbes. It's the most current general consensus. As we get more information, we change our minds, names of bacteria change, we move them into different groups, and that sort of thing. Now, previously, when we identified and classified organisms, bacteria in particular, when we were putting into taxonomic categories, class, phylum, genus, species, that sort of thing, we went with shape, whether they had flagella, where the flagella were, how they reacted to various staining protocols, biochemical tests, etc., because we didn't have anything else to go on. They only come in about 6 shapes. When you're looking under the microscope, there's not a whole lot to go on. So we went on with what we could. Now, recently we have had improvements in our DNA technology, and we have been able to organize the bacteria according to relatedness. So there's been big shakeups in the taxonomy of bacteriology, and the current Bergy's Manual, from which this textbook is based, tries to follow that most current classification. So currently we try to go by DNA sequences that suggest relatedness, then we go with shape and all of these other things. Because of that, we now have very different looking taxonomy for prokaryotes. First off, we have a new domain, Archaea, of which we have 3 phyla so far within Archaea. One of them only has one poor little species. I'm sure we'll find more. Bacteria, on the other hand, we have several phyla, and they're somewhat different than how organisms were organized previous to this. Like I said, the textbook follows the most current consensus. So here are reminders of what you should have learned from the previous 2 topics. On general characteristics of prokaryotic cells, here's the learning objectives for that. And for classification of prokaryotes, here is a reminder of what you should have learned from that topic.